thank you for joining us for the fifth Ocean Nexus webinar. This event is presented by Nippon Foundation and Ocean Nexus. Nippon Foundation, some of you wanted to know, is an international philanthropic organization promoting actions to achieve a society in which all people support one another. So Ocean Nexus is supported by Nippon Foundation, and we organize a webinar with their support and with the colleagues' support and participation from Nexus Network. So Ocean Nexus, us, is a research network of scholars who conduct their research with empathy by employing anti-racist, decolonization, and intersectionality as our analytical lens. As a group, our mission is to establish social equity at the center of ocean governance. My name is Yoshitaka Ota. I'm the director of Ocean Nexus, and I'm a professor at the University of Rhode Island. I'm the host of this webinar. Not really that good one, but I'll try my best to decipher this really complicated topic that we are tackling today. So today's webinar, Environmental DNA for Oceans, White House Strategies, International Standards, and Critical Perspectives, is part of the Ocean Equity Learning Forum webinar series, focusing on the research theme, data and digital technology for oceans. So I'm so pleased to have three eDNA expert speakers joining us. Our first speaker, Dr. Elaine Shen, is a 2024 Sea Grant Knaus Marine Policy Fellow in U.S. National Science Foundation Division of Ocean Science. Elaine earned her PhD in biological and environmental sciences from the University of Rhode Island, yay, and BS in ecology and evolutionary biology from Rice University. Her research has drawn upon a variety of disciplines to examine the social and ecological dimension of marine biomonitoring, with a primary focus on environmental DNA. Okay, so I'm so pleased to have you as a speaker, Elaine, and thank you for joining us. And we're just gonna go. Our second speaker, Dr. Amelia Moore, is an associate professor of marine affairs in the College of Environment and Life Science at the University of Rhode Island. She earned her PhD in sociocultural anthropology from the University of California, Berkeley, and her BA in environmental biology from Columbia University. Got a long side of things to say, but Amelia's approach, her research areas through the adaptable lens of feminist studies of science and Black ecologies, building bridges between critical theories of social differences, anthropology, anti-racist and anti-colonial studies, political ecology, and socio-ecological sciences. Amelia, thank you very much for participating in this, and well, thank you for bringing me up, to Rhode Island too, again. Okay. Right, and our third speaker, my wonderful colleague, Dr. Shana Hirsch, is an assistant research professor in the Department of Human-Centered Design and Engineering at the University of Washington. Yay. She's also co-director of Pacific Marine Energy Center, where Shana collaborates with industry, government, and university researchers to understand innovation in marine energy. She's university researchers to understand, and university researchers, I'm so sorry. Shana is also a research scientist at eDNA Collaboratives, and she has an interdisciplinary background in sustainability, social science, and water management and policy. And Shannon and I have been working for the last 4 years. And so I'm just so glad I'm bridging time like a friend back in the University of Washington and new colleagues in University of Rhode Island. So it's my pleasure to host this. So first, I'm gonna ask Elaine Shen to introduce, federal perspective on eDNA and its alignment with other kind of like a Biden-Harris ocean policy priorities. It's a very typical, important topic right now. And then I'm gonna ask Amelia to kind of have a reflections and understanding what can we say about eDNA from critical perspective, because Amelia and Elaine published a paper together, and then I want Amelia to promote the paper so everybody read it. And then I'm going to turn around to Shana and ask her to explain it. Okay, so now we talk about like eDNA, but what about like expanding eDNA? What about the application of eDNA in global south? So we're going to talk about the global, what's the concern, what's the implications. I'll ask Shana, based on her new publications, everybody should read it, again, to give us a talk. So enough about me. I'll pass it to Elaine. Elaine, please take the floor.
Okay, thank you so much, Yoshi, for that lovely introduction, and hello to you all. Hopefully you can see my slides now. Please let me know if you can't. I took a quick look at the webinar registration for this meeting, and I saw a lot of different time zones represented. So I would love to hear where everyone is from in the chat, and yeah, feel free to introduce yourselves in the chat. We'd just love to see who's on the call with us today. But like Yoshi mentioned, my name is Elaine Shen, and I'm currently a Knauss Marine Policy Fellow in the National Science Foundation's Division of Ocean Sciences, and previously did both ecology and social science research related to or using environmental DNA. So you'll hear a little bit more actually about some information for us to discuss further. Did my internet just go a little unstable?
Yeah, you froze a little bit. Can you back up like 30 seconds? Is that okay? I know you're not Netflix, but you know, you can back
Sure.
up a little bit, you know, that'd be really great. Everybody okay with this?
Yeah,
Thanks
happy
very
to.
much. All right, thanks so
Happy
much.
to.
Sorry about this.
Yeah, it's a rainy day here in DC, so I think the internet is a little bit unstable, so my apologies on that. But I was just saying that, you know, really what I'm here to provide is some of the policy context that I've gained in the Knauss Fellowship, which, yeah, again, like I didn't know very much about when I first started, but might be familiar to some of you. And really, I'm here to provide my perspective as a fellow and as an individual. And so not really speaking on behalf of the US federal government or really any of the groups that develop these documents, but really to help set the stage for this conversation. So really looking forward to it. And so here's an overview of what I will be covering. So I'll first briefly introduce here what eDNA is and what it's been used for. And then I will provide some quick details about the National Aquatic eDNA Strategy, and then end with some alignment of the National Aquatic eDNA Strategy with other Biden-Harris administration ocean policy priorities. And I know that we have some folks from international contexts here, so really I'm providing kind of that US policy perspective, but I think a lot of what we're going to be discussing here today obviously has more far-reaching implications. So first, what is environmental DNA? So simply put, eDNA or environmental DNA is the genetic material that's found in the environment, including in fresh and saltwater ecosystems. eDNA can come from a variety of sources through naturally occurring processes that organisms undertake every day as a part of their everyday life, like the production of mucus, the shedding of skin cells, waste, decaying matter, and even gametes. And the DNA itself can come in a couple of different states, either intracellularly, so inside the cell, like dead or live intact cells. And it can also happen or be collected extracellularly. So this includes eDNA fragments, those that are particle bound, and those that are partially dissolved. And so eDNA can persist in aquatic and marine environments. Studies show somewhere on the order of hours to weeks, depending on the environmental processes in those environments and the state that it's in. And to utilize eDNA, there are a couple of general methodological steps that every protocol has. And so first, after you collect that water or fresh, that water sample, you would concentrate the DNA from that sample through a filtering process of some kind. You'd then go into the molecular lab and extract the eDNA using specific kits and amplify them using PCR or polymerase chain reaction. And then finally, you translate those physical DNA molecules into digital ATCs or Gs using a high throughput sequencing machine. And once you get those digital files, you can then analyze the sequences bioinformatically to ascertain what DNA sequences belong to what organisms. And more specific considerations for each of these steps are possible depending on the research question being asked. And so what this methodology and eDNA allow for is a more than meets the eye approach to censusing or detecting biodiversity in marine and aquatic environments. And so by relying on the DNA that organisms naturally shed or produce into their surrounding aquatic or marine environments, eDNA can help detect invasive or endangered species. It can help surveil harmful algal blooms, evaluate environmental impacts and restoration efficacy, can help assist in maybe more ecosystem-based fisheries assessments, as well as other uses more generally related to biodiversity monitoring and ocean exploration. And the reason why, eDNA has been really exciting for folks and has really seen a lot of development in the past, I would say, decade and a half or so, is because of this increasing demand and need for biodiversity data that various actors are looking for to better inform decision-making. And so it's not just the US government, which I've abbreviated here as USG, who has increasingly pressing needs for reliable information on the nation's ocean species and habitats, but we're also seeing those needs expressed by the private sector as well as in international fora. And one of the challenges really here, I guess, in the more US context in addressing this need for biodiversity data is that a wide range of actors in the ocean life sciences community, maybe even some of you on this call, are currently working on eDNA, have and generate that kind of information, but that data is currently variable in quality, not well coordinated, and oftentimes inaccessible. And so managers, decision makers, and other interested parties in the ocean space are really asking, again, in the US context, for this federal coordination and leadership to meet this demand. I'm going to pause for a second because I see some stuff in the chat. Oh, great to see introductions from all of you. Okay, so the National Ocean Biodiversity Strategy and the National Aquatic eDNA Strategy aim to address these needs and knowledge demands in a complementary fashion. And I'm presenting these both to you because they were released on the same day earlier this year in June. And really what both emphasize is the need for improved communication and coordination to provide trusted biodiversity observations and metrics across mission-driven federal agencies, academia, industry, and philanthropy. And so while the National Ocean Biodiversity Strategy provides a broader picture of ocean biodiversity science and information, the National Aquatic eDNA Strategy really focuses on the eDNA methodology and technology more specifically and addresses some of those more technical aspects. Let's see. And so since we're here to mainly talk about eDNA, here are the goals of the National Aquatic eDNA Strategy. So its first goal is to coordinate across sectors to facilitate integration of aquatic eDNA into decision-making. The second is to build a human capacity infrastructure and research enterprise needed to employ eDNA technology at large scales. And third, the third goal is to advance coordinated aquatic eDNA observations to aid comprehensive assessments in US waters. Some of the key intended outcomes of implementing the National Aquatic eDNA Strategy include harmonized technical approaches and strategic commitments in capacity and infrastructure development for priority applications, the coordinated observations and metrics for the unprecedented characterization of aquatic life, and aligned communication strategies to increase public confidence and trust in environmental assessments. And so achieving and implementing towards these outcomes will be crucial to meeting some of the US administration priorities, and specifically its ocean climate priorities. And so here are a couple of key examples of high-level ocean policy efforts in the US government for which better and more coordinated biodiversity information are envisioned to improve decision-making processes. And so here you'll see screenshots of the Ocean Climate Action Plan, the National Strategy for a Sustainable Ocean Economy, and the Ocean Justice Strategy, which are all, again, complementary strategies that at a high level aim to protect the ocean effectively, produce from it sustainably, and benefit from it equitably. And so very briefly here, because I don't want to run too much over time, the Ocean Climate Action Plan, released last March 2023, provides actionable recommendations for the US federal government to achieve ocean climate solutions. The National Strategy for a Sustainable Ocean Economy, which was actually also released this past June in 2024, considers how the US can sustainably manage oceans under its national jurisdiction and provides that whole of government vision for resilient, prosperous, and secure ecosystems, people, and communities. And then finally, the Ocean Justice Strategy, which was released in December of 2023, so December last year, provides a vision for how ocean justice derived from the environmental justice movement can be incorporated into federal decision-making processes related to the oceans. And so now I thought I'd provide maybe a more concrete example of how eDNA might help achieve some of these ocean policy priorities through how it can help determine the ecosystem impacts of various ocean climate mitigation strategies, since these emerging or existing uses are happening and affecting ocean ecosystems at the same time. And so really, I think if done right, eDNA can provide the biodiversity information needed for communities to have a greater understanding of how their ecosystems are being impacted. And in return, also, communities can identify geographic areas or industries or other concerns or priorities of interest in their coastal environments in a manner. And let me pause. And what this strategy envisions really is that the federal government can organize in a coordinated fashion to meet those community needs. And more importantly, really, is to leverage taxpayer dollars effectively so that we don't duplicate efforts, especially for approaches that are unsuccessful. And so finally, I just wanted to leave you with 4 take-home messages. The first being that eDNA is a powerful and emerging tool that can help address growing needs for biodiversity data and monitoring ecosystem change. Second, that the National Aquatic eDNA and Ocean Biodiversity Strategies address the growing needs for federal coordination and leadership that has been requested by a wide range of constituencies. Third, that successful implementation of these strategies, which is ongoing, will help the US deliver on its ocean climate, sustainable ocean economy, and ocean justice goals, as well as a range of existing federal mandates. And finally, and this is where many of you come in as the audience, federal and non-federal leadership, creativity, and partnership are really needed to establish mechanisms towards successful implementation. And so I'm going to stop there. Thank you all so much for listening. And I will turn it back over to Yoshi to introduce or hand it over to Amelia.
Thank you very much, Elaine. It's a wonderful overview, and it's quite clear that there's layers of strategies that connect to each other, and it just notes how important it is to strategize as much as coordinate. But I suppose when it comes down to, so I'm going to be honest with you, to resilience and sustainability and the new technology and sustainable ocean economy, somehow somewhere inside of me, well, to what extent is it possible? And then when it comes with words such as justice, it just sort of like a ring a bell to me. Well, some warning to it. How much we can rely on it, and what do we need to be careful about? So I know that you published a paper with Amelia, and I'm going to pass it to Amelia, and then Amelia, my concerns and perhaps some would wonder, is it like right things to think about, or how can we actually take this new emerging technology to be matched with our advancement of equity and justice in the oceans. What do you think?
Thank you, Elaine. Thank you, Dr. Ota. I am going to share my screen successfully, imminently. Here it comes. And in a second, you should do slides. Any second. How's it working? Okay, good?
Great.
All right. All right. So this is just a continuous conversation about eDNA and its relationship and connection to equity and justice issues locally and globally. And this is a kind of a broad overview, a bit that's based on the paper that I hope you will engage with at some point, that was written by Elaine Shen, Jessica Vandenberg, and I. We collaborated on this paper to highlight our concerns about the socially uninformed use of eDNA as a basis for environmental policymaking and resource use and conservation. So this paper stems from, really importantly, from a chapter in Elaine's dissertation about eDNA with insights from political ecology and environmental anthropology. So what we don't talk about enough when we talk about tools like eDNA, especially in the labs and in the published papers where a lot of this technology is actually developed and the details are discussed, we don't talk about the fact that technology is never neutral. It cannot be understood outside of its social and political context. So as a science studies scholar like Bruno Latour has famously said, the context and the content are always one, are always the same. And of course, this includes biosensing technologies. We do often talk about eDNA's capacity and our desire to enhance its capacity to inform ecosystemic impacts, climate actions, and the development of the ocean economy, as Elaine just showed, but we must also delve deeply into the potential and likely justice issues that suffuse this technology with ethical implications and inequitable effects. So we need to consider long-standing injustices, such as ecological imperialism, for example. This term references the fact that most ecological and conservation science stems from the developed world, particularly the Euro-American former and current imperial and colonial powers. In the supposedly post-colonial era, ecological and environmental science and conservation biology and related policies at multiple scales, from the global to the local, have become a more socially acceptable means of, some would say, accumulating resources for neo-colonial powers and industries. So what this means is that instead of accumulating raw materials and land, what is imperialistically controlled is now access to information, what counts as appropriate and legitimate knowledge, relationships with local ecosystems and the localized concepts and languages that stem from those relationships, and the authority, like who controls the uses and meanings of biodiversity. And this can lead to communities and local populations becoming dispossessed of their environmental connections and key relationships by means other than the violent removal or theft. In other words, biosensing can be imperialism by other means. Another really important concept that I don't think we talk enough about yet is data sovereignty and how uneven data sovereignty is around the world. And this refers to quantifying, categorizing, ranking, and otherwise transforming local place-based generational relationships between people and the complex ecologies in which they coexist, transforming all those relationships into data. Some people call this datification. In many cases, certain forms of data usage can make it impossible for some people to participate in the management of their own living systems. Such transformations can break or erase long-standing ways of knowing and being with reductive forms of accounting that are often very blind to history, to social context, to identity, to other forms of local meaning, and to forms of mutual interdependence that people have with their surroundings. And this can make it difficult or impossible for people who utilize other ways of knowing or other forms of accounting, especially indigenous communities, to retain autonomy over policy decisions based on forms of data about people, about land, water, resources that they don't recognize as meaningful, or data that excludes them altogether. Additionally, forms of data collection that require prohibitively expensive or technologically complex tools can prevent inclusive participation by communities, municipalities, or even whole countries who do not have access to those high-tech forms of data generation. And those kinds of disparities can also prevent autonomy and sovereignty over data collection, analysis, interpretation, and utilization. And then surveillance. And by this, I mean the capacity for powerful actors and decision-making bodies to collect information about places, ecosystems, populations, and practices at a distance, and to make policy decisions for people and places without deep place-based relationships or deep connections to the affected areas that are being surveilled. In this context, then, eDNA can be used as a tool for the evasion of consequences for unpopular outcomes and for the capacity to ignore any detrimental local effects of resource management decisions. It can also be used to artificially separate data from its local place-based context, enabling the removal or erasure of affected populations from policy considerations and other forms of recognitional justice, allowing powerful entities and actors to benefit from this information without benefit sharing or consent. So our point is that inequitable power dynamics such as these can exacerbate and inflame global, regional, and local hierarchies of knowledge production. We contend that these considerations and concerns are human dimensions of great importance for eDNA research. So some key takeaways, and again, we hope you will have read our contribution to the conversation or that you will soon, but some key takeaways in case you just want a little cheat sheet, is that the critical social sciences and science and technology studies in particular have a lot to contribute to our understanding of the ethical utilization of eDNA. eDNA scientists, practitioners, and decision makers hoping to utilize this technology should become far more familiar with such critical scholarship as part of their fundamental training. This should not be optional. These arguments should not be marginal either. Another key takeaway is that representational rhetorics matter. And here we refer to the work of Paige West in her seminal book, Dispossession and the Environment. We need to be careful how we communicate about the value and uses of eDNA, because the way technologies are described, framed, and valued affects how they are deployed, who is or who is not included in decision-making about that deployment, and how data collection is interpreted, used, and owned. So in an analysis of recent literature about the scientific relevance of eDNA that was conducted for this paper, we learned that common representational rhetoric about eDNA does not often address social inequities or uneven power dynamics between those who control the deployment of the technology and those whose lands and seas are surveilled by it. Instead, the rhetorics of eDNA tend to promote the neoliberal insertion of international capitalist markets and the private sector into environmental management. Also, the celebration of technological solutionism, the idea that most complex problems can be solved by more data and more efficient technology at the expense of more transparently equitable solutions. Also, this assumption of molecular genetic reductionism as being preferable, the idea that living relationships are best understood or somehow more true at the genetic level. All of these framings and assumptions can actually exacerbate systemic inequities when they discount or erase marginalized populations and communities from the picture and or redirect accountability away from historic and contemporary sources of widespread ecological degradation, such as industrial pollution and extraction stemming from very imperial, colonial, corporate economies and actors. The more mainstream rhetorics of eDNA that we're aware of tend to favor global hierarchies of knowledge production, in which the global north has more technological and data processing power than the global south, and top-down implementation strategies in which the technology is deployed to surveil wide swaths of planetary space without input, or sometimes, all too often, even the awareness that this is happening from affected populations. And these disparities can erode more equitable forms of communication and collaboration over biodiversity conservation and environmental management. So to date, discussions of social equity and historically mediated uneven and asymmetrical power dynamics are not often prioritized in mainstream conversations about the efficacy of eDNA technologies. And I just want to mention some recommendations. We can come back to this. But we want to center, like these are stemming from the paper, we call for centering the rights and sovereignty of local and indigenous communities affected by eDNA surveillance. We call for the institutionalization of consent-based eDNA research, really looking for that informed consent, utilizing fair and care data principles. And if you're interested in that, there's the title of the paper by Carol and others that came out in 2021 there. Invest in local means of eDNA knowledge co-production with local communities and populations, and reflect on your own positionality as eDNA practitioners, scientists, and we can come back to that too. So I will end there for now, and I hope to get into some of this more in conversation.
Thank you very much, Amelia. That's powerful messages and clear kind of like a interpretation of what we should actually think about it, efficacy of eDNA. Stick to my mind, and it became very much central. And so I'm going to pass it to Shana, and Shana is working to re-implement the eDNA capacity and increasing capacity in the global south. And she's written a paper, as I said, and really just thinking about efficacy on the one hand and capacity building on the other. So Shana, would you actually be able to expand this conversation by going through your paper, please?
Yes, thank you so much. And thank you so much, Amelia. I hope that this paper really speaks to a lot of the really important issues that you brought up. My name's Shana Hirsch, and I am working as a research scientist and assistant professor at the eDNA Collaborative, which is a team of researchers at the University of Washington. We're working to support eDNA research around the world and bring the tools to people, especially in the global south. My job has been to really support the needs of researchers in the global south. And I'm going to introduce a recent paper that was just published yesterday. And it was written by a team of researchers who I know some are also in the call. Yay. And I will introduce you to our findings. So scientific standards, let's see, let me get this going. Okay. Yeah, okay. So scientific standards, as we've heard, are an important aspect of ensuring quality and reliability for science. They're often essential for developing trust in a scientific enterprise. And standards can really range from ad hoc and informal standards, best practices, to those that are developed by official standards bodies, such as the ISO, or the International Organization of Standardization. And they also can include both international and national standards. So right now, there are several national efforts that are aimed at creating standards for eDNA. And there are also some nascent efforts to develop international standards through the ISO. But the standardization effort has largely been driven by researchers in the global north, from countries with some of the greatest access to state-of-the-art technologies, laboratories, and funding. And those countries include Europe, North America, Japan, and Australia. So this really skewed access to the standardization process is really originating from these inherent inequalities in access to infrastructure and funding and technology. And they could perpetuate some of the things that Amelia was speaking about, and they could perpetuate unequal participation in eDNA research in the future as well. So we believe that for eDNA technologies to be accessible, we need to have a broad conception of users from around the world and consider all of their different contexts and locations and abilities to engage. And we also need to really consider, building on Amelia, the social and ethical implications of eDNA standardization as well. So to work towards engaging with the standardization process more fully and bringing in voices from around the world, we assembled a group of people from around the world to gather their perspectives on international standardization of eDNA. And these folks are from Africa, South America, Asia, Pacific Islands, and other parts of the world. And we brought in 18 researchers to develop a manuscript collectively. And like I said, some of these folks are in the call now. The goal was really to understand how efforts to develop international standards might support or raise concerns for researchers using eDNA methods and technologies in the Global South, and also in remote and difficult to access areas. So we used a series of open-ended questions and an iterative writing process where we are all co-authors, and we gathered these perspectives together to present in a journal to an audience of folks who are concerned, who might be making and creating international eDNA standards. So the results are included in detail in the manuscript, but I'm going to outline some of those here. So in regards to the current use of standards, we found that there really wasn't a universal definition of standard, although most participants used some form of standard in their work. And that could be either ad hoc standards or adapting national standards to meet their own needs. We also found that in regards to benefits from international standards, every participant identified benefits, either from existing standards or from potential ones. And this included several points around ideas about reproducibility and collaboration, guidance for research, increasing credibility of eDNA. And some participants really wanted to have a common nomenclature and clear guides that would help them develop workflows for sample collection through bioinformatics and data reporting. And this would, in the view of many of the participants, this would make eDNA methods more accessible to researchers across the world. So although there were really differences in emphasis and opinion, many of participants also aligned in their concerns about international standards creation. And these concerns centered around resources, including cost, accessibility, and capacity, questions about the technical feasibility of standardization, and the data requirements and ownership. So in terms of access and capacity, access to resources that are necessary to meet standards are often difficult to come by. So these resources could include finances, access to technologies, materials, equipment, as well as capacity for personnel to run a sequencing machine, for example. And participants were really concerned that if the cost was too high, the standards would be out of reach and impossible to achieve, making them elite. Fundamentally, access to materials and technologies is a major barrier to meeting standards around the world. And this is especially true if capacity and funding are already stretched for researchers. There's difficulty accessing state-of-the-art equipment. Laboratory sterilization is also a particular problem. And there are really entire regions of the world, such as polar regions, Amazon forest, for example, and situations such as on research vessels where there's no sterile extraction lab. And so any standard that required one would be unattainable. There are also technical concerns. Many participants were really afraid of a one-size-fits-all standard or protocol, and that there really needs to be a range of sampling designs, data analysis, designs for different locations and taxa and ecosystems, and that different eDNA technologies are also at very different stages of technology readiness depending on the application. Some participants were also concerned that standardization at this stage would maybe stifle innovation and make it difficult for the field to change in the future. And then also there was concerns around data. So much as Amelia suggested around and spoke to around data sovereignty, data sharing and standardization were another set of concerns that were shared by almost everybody. Beyond the technical aspects, people were concerned about data ownership and control, particularly because this is in relation to genetics. And there's no precise, there wasn't really a precise consensus around from all of the participants and the authors, but people discussed generally the legacy of genetic colonialism and the issue of colonial science more generally. and that there really needs to be a real notion where people are understanding data security in a really specific way, and that this is related to cultural and personally sensitive data. And it's especially true when this is a culturally important or threatened species, which could be subject to exploitation without community consent. There could be overharvest, and many people did not want the data on these species to be geolocated. So to address these problems, some themes arose. And I think these themes really align well with the findings from Elaine and Amelia's paper as well. So one of them is to create adaptable standards. So there's really this common perspective that we need pragmatic guidelines to make research comparable, but not so stringent that some researchers are left behind. And this may seem strange because flexibility may seem at odds with standards, but really the standards are acting as boundary objects. They're enabling people to have shared meaning and communication across communities. And this is especially important to consider in terms of communication to regulatory bodies and developing a shared trust concerning this new form of environmental data. So in this vein, standards could be created and reported in ways that ensure adaptability by taking into account those things that are minimum requirements, like need to have, versus ideals or nice to have. And this would allow more people to use standards and speak across locations and fields and applications and regulatory bodies, especially as the technologies are in flux. Second theme was to develop regional capacity. So one overarching desire is the need to strengthen regional or in-country capacity for eDNA research. And increasing regional capacity will help avoid imbalances in parachute or colonial science and enable researchers in the Global South to have more access to eDNA technologies and capabilities. So one example of this is the Open Institute of Africa Biogenome Project. And this is an excellent example of a consortium building regional exercise, which is building capacity and offering regional workshops and knowledge exchange within Africa. And to date, the project has trained over 400 African scientists in hands-on molecular biology and genomics, and they've engaged over 3,500 African participants in regional workshops. And regional hubs or consortia could help resolve some of the issues relating to genetic data and genetic property by keeping samples local, and regional networks can help produce libraries and methods that are relevant to particular localities and ecosystems. Third, we need to sensitize to data sovereignty. Standards should be developed in a way that provide options, realizing that not all data can be geolocated, and some researchers and communities may want to opt out of that entirely. And they may not want to publish sensitive data due to issues of sovereignty and security. So beyond data, eDNA standards should consider ethical engagement with indigenous communities more broadly and include these existing frameworks, which include the CARE principles for indigenous data governance. And these are already being incorporated into institutions such as the Smithsonian Institution and others, and they could be adopted by all genomics organizations and incorporated into standards. Finally, the fourth theme was to view standardization itself as a global capacity building exercise. So although international standards have no technical requirement to be attainable by all, if a standard is truly international, it should be able to be used by everyone around the world, and they should have the capacity to be able to meet it. So in other words, while it's important to have a standard, it's more important that everyone can use it within their own situations and contexts. So the goal of standardization should be a process-based goal where we're building capacity as we're standardizing. And importantly, rather than hindering development, dialogues taking place around standardization of eDNA methods and technologies could act and should act as catalysts and democratizing exercises. But in order to do so, this dialogue has to take place across all hemispheres of the globe and invite a wide range of stakeholders into the conversation. So Finally, I would like to thank again my co-authors who worked very hard on this paper, and also to the funders, David and Lucille Packard Foundation. I encourage you all to check out the eDNA Collaborative at our website, download our article, or send us a message if you would like to find out more. Thank you all very much.
Thank you so much, Shana. It's really interesting. There was one question. What's the difference between standard and protocol? Would you be able to speak very quickly about that?
Yeah, so standards is kind of a broad term that can include protocols, and protocols would kind of be a workflow, and a lot of standards include a particular protocol. So I hope that that helps. It's like a nested thing.
Okay, thank you. Nicole, is that okay? Right. So I'll pass it to, I want to ask Amelia, come back to your recommendation because you went through very quickly. Nicole says yes. Okay, great. So okay, so you went through very quickly. I mean, in your paper with, Elaine, Amelia, you guys talked about intentionality, and you also talked about positionality. That really was very important part of your paper to really warn us, there's something that we really need to consider when we apply new technology. Can you just speak a little bit about both intentionality and positionality too? And those who ask a question, I'm going to pick up a few and then I'll ask them later, yeah? So Amelia or Elaine, please.
Maybe I'll start and then Elaine can follow up if you feel like it, Elaine. But I think we always should be pushing understandings of our own positionality within technological assemblages and contexts. We all have different roles. We all come at it from different perspectives and places and angles. And that has meaning and consequences. So ask yourselves, how did you come to the eDNA field? How did you get here? What ethical considerations guide your work? What communities and populations are you accountable to or responsible for? What social and political contexts do you assume are external to your work versus what do you assume is vital to your work? And who directly benefits, and this is critical, who directly benefits from the outcomes of the projects you participate in most immediately? Who directly benefits? And who is excluded or marginalized? And how do the projects that you participate in enable or constrain or prevent legacies of marginalization and inclusion and exclusion from being perpetuated? So these are all things that I think should always be top of mind when we're doing biosensing work like this, because it has such potentially profound consequences to perpetuate inequitable legacies. And I think we all have to just be aware of why we're involved, who we are, who's benefiting, who's excluded, etc. Elaine, did you want to add?
Yeah, I'll just briefly mention maybe a concrete example speaking for myself. So when I was writing this paper with Amelia and Jess, my positionality was very different than where I find myself now, where I'm in mostly federal spaces trying to think about, now that there is a national aquatic eDNA strategy, what is the federal vision for how we can implement it, and what kind of recommendations are legible to other decision makers that I'm with. And so it's important for me to kind of wear different hats and leverage them in different ways, and think about kind of how power dynamics shift even as I, as an individual with like my unique experiences and with my unique experiences move in these different spaces as well. And I'll add that, I would love to hear more speaking to this question from Shana, actually, on how you actually convened all these people from all over the world, and maybe some intentionality in that process.
Shana, do you want to answer to that, please?
Yeah. So this was one of the most rewarding and fun and challenging papers that I've written. So I really sat down and I said, okay, we need perspectives of people from around the globe who are doing eDNA research, and we want to hear from them. What would be the best way to engage with them? And I've done a lot of interviews and kind of large scale ethnographic work before. But when I engaged this time with folks from around the world, I really wanted to have them be co-authors and for us to write this this paper together. So I helped facilitate that by asking a series of open-ended questions and then drafting subsequent drafts and kind of going back and forth with the co-authors as they added their own opinions. And this was actually really interesting too, because the audience for this paper is a very quantitative journal that's a biosciences journal. And so there was also a lot of negotiation around you know, the what evidence would count and how we could present qualitative perspectives in a biosciences. So I also hand it to the editors for that journal for being open-minded about that.
Gina, I want to ask all three of you. So first of all, we're talking about eDNA, but we're talking about eDNA in ocean. You know, how advanced the technology will be. I mean, it's very difficult to just really know what's in there when it's sort of like a fluid, such an environment. I mean, that's the first thing. And if not, you know, what is the risk out there? So, you know, in my mind, it's simply there is the contesting data between what eDNA presents and what the local community presents. When somebody comes in and decided whether we're going to create a protected area or not, you know, if there is a conflict between those two, how we can decide which one is the right information? You know, how can one community who doesn't really have access to the capacity of the eDNA can kind of like, you know, argue against what eDNA says? I mean, in fact, is it really so like so solid that we actually know what's out there? I mean, that is kind of come to me. I mean, it's great to know what's out there through the eDNA. It's absolutely brilliant. I mean, that's what the scientist wants to. We want to know more, right? With the new technology, but how sure we are that we can actually manage those risks, you know, considering the inequity and the power imbalance that we have right now. What do you guys think? Or is it like too much to ask?
I have some thoughts. I mean, I think that any sensing technology, including our own eyes and observation, is going to have risks. It's going to have trade-offs, and it's going to have faults, and also could potentially be wrong, right? And so I think that right now we're on the cusp of the technology being accepted and trustworthy if we can develop these kinds of protocols and capacities around the world to make sure that the science is being developed and used in a trustworthy way. But I think that, you know, if you're going out and capturing, say, fisheries trawl data, you can do, you know, you are not going to catch all the fish in the sea with a trawl, right? And you're not going to, the same thing with eDNA. So it's a matter of finding, you know, the relative risks and proportionality between what you are and are not capturing, just like any other sensing data.
Thank you. We have a few questions, so I'm going to go very quick. We only got the 5 minutes, but I always like to just read out those questions. So Dr. Elaine Fasterman, our collaborator from University of Washington, asked, okay, so is there what's been done with open maps of indigenous resources? I think that's a very heavy question. I don't think there has been that much good things has been done, really, and it's very difficult to say what's indigenous resources and what's not. But does anybody, you know, perhaps Shana, what you introduce in consortium in Africa, it would be a good example for the Elaine to have a look at it. I mean, you know, Shana, if you can just put it on the chat, that'd be really great. Thank you so much. So I'm going to go really quick. I'm so sorry. You can just, you know, and Joe, you asked, it's really interesting. You said you can do this eyewitness survey and Rio and eDNA. Is that too much? Elaine, would that be too much?
I would say if you're ever doing an eDNA project for a dissertation, to always have a plan B, just as a short note. But I think that if you have the data that you're describing already, then it is not too much for a project, and it's actually good to have that as sort of other data sources for a particular project in case the eDNA doesn't produce the signals or the quality of data that you need. That's something I experienced in my own dissertation, which is why I mention it. Thanks.
Thank you. Brian, thanks for your comment on USG, and then they honor communities. And, I just want to ask you back, do they really think about the positionality, intentionality, when they're actually asking the process of it? To ask, whether indigenous community wants to use it or not. It would be great to have a conversation sometimes. But you have a question to the all panelists. Who has access to the eDNA data collected by USG? Does anybody know?
I think there are actually a couple of NOAA colleagues who might be on the line who might provide this information more directly than me even. I've been able to work with some eDNA practitioners who work for the federal government, and I think there is a strong desire to have, a central repository that is open and accessible for eDNA data, but that currently is either in development or exists and I'm not aware of it yet. But that is one of the goals of the National Aquatic eDNA Strategy, is to have that coordinated information in one place so that users can utilize that information and products can be derived from it for easy access. So sorry, that's a very quick answer because I know we're short on time.
Yeah, Brian, I hope that's okay. Somebody else asked, eDNA collaboratives at UW provides trainings. Yeah, just Google Shana and Ryan Kelly, who's my good colleague, and you can ask them. And Pedro, you put really interesting, or important comment. Collaboration is something, you guys value greatly, but we often find ourselves in second or third place in these partnerships. And I think it's really, it's a real voice. What strategies or initiatives can be implemented to ensure equitable access to these technologies and to enhance capacity building for researchers in developing regions? How can we ensure that our voice of prioritizing in these efforts, especially if funding comes from abroad? I absolutely agree with you. That's a real voice to it. Thank you very much. I'm hoping our webinar continue to discuss about this and can really answer to your question in future. And, thank you very much for joining us. I can only say this, but, let's keep in touch. And lastly, Adrian, yeah, how could they join? Okay. That's a question to Shana, isn't it? And we're trying to just put the paper as much as we can. Okay. That's pretty much it. If you have any more questions, please contact to Ocean Nexus, and then we're just gonna ask to our panelists. And we got about like a minute now. And I just wanna thank you everyone. And I know some people attended from mid of night and really appreciate. And thank you very much for all our panelists who's just really talked passionately and in such a current topic. And thank you very much for organizing this, Ariel and Karin. And thanks for all these partners and participants again. And have a good night, good day. And thanks a lot. See you next month. Bye.
Yes, please reach out if you have any specific questions. eDNA Collaborative. Yeah. Thank you.
Well,
thank you, Jenna. Thank you, Amelia. Thank you, Elaine. Thanks, Ariel. Okay. Thank you, Oma. Thank you, Brian. And somebody was from Philippines. I think it's past midnight. Thank you so much. Dr. Firstman, thank you very much for your participants. We have somebody from Nigeria. Thank you. And Mokoro, thank you very much for this encouraging chat. It helps me to have some little confidence. Thanks, Brandy. Thank you, Jason. Okay. And anybody who I didn't call, thank you so much. Okay, now we're gonna close.